Greetings, everyone. Morning, afternoon, evening, wherever you are in the world. Thank you so much for joining us for the Global Foot and Mouth Disease Research Alliance, GFRA's webinar series. We're happy to have you with us today. We just have a couple of comments, some housekeeping items, and then we will turn it over to the presentation. During the Zoom webinar, you'll see the Q&A box down at the bottom of your screen. Please feel free throughout the presentation to write your questions in this box. And then during our discussion period, our moderator and presenter will discuss your questions and really spark a good discussion around the topic. I would also like to plug for everybody the GFRA scientific meeting coming up November 8th through 10th of this year in Kampala, Uganda. We're very excited for this meeting to take place, and we'll be sending more information and a link to register for the meeting in the chat shortly, and we hope to see all of you there. I will pass it over to Dr. Giselle Medina.
Good morning, good afternoon, good evening, everyone. Thank you for joining today's GFRA webinar. It is my pleasure to introduce Dr. Anna Jolles. She's a professor of epidemiology in Oregon State University. She obtained her PhD in ecology and evolutionary biology from Princeton University, followed by a postdoctoral work in the University of Groningen in Netherlands. Since then, she's held various faculty positions at Oregon State University. She's a professor, and her position is split between the Carlson College of Veterinary Medicine and the Department of Integrative Biology. Her research is focused on the study of infectious disease processes in natural mammalian host populations. This has included many different parasites and pathogens in African buffalo in South Africa, and more recently, desert bighorn sheep in California. The work that she leads often bridges organizational scales from cellular immune processes to disease transmission in host populations. And her research group uses observational and experimental field studies, as well as mathematical models to address specific questions. Today, Dr. Jolles will present her research titled Multi-Scale Dynamics of Foot-and-Mouth Disease Viruses in African Buffalo. So without further ado, Dr. Jolles, the floor is yours.
Thanks, Giselle, for the introduction, and thanks for having me. It's a real pleasure to have the opportunity to speak in this forum. So let me start my screen share here. Okay, is that showing up?
Yes, it is.
Okay, great. Then I'll get going. I've had a lot of caffeine because it's 6 a.m. West Coast time. So I'm going to have to try and speak like a normal person. Yeah, so I'll talk to you today about multi-scale dynamics from cells to landscapes in African buffalo. And of course, that is not truly just my talk. It is, there we go. It is, in fact, a really large collaboration, and the people I've listed here are just the PIs and postdocs on the project. There's also a small army of graduate veterinary and undergraduate students who are participating and have participated in this project. So what we're trying to do really is to connect infectious disease dynamics across organizational scales here. And in the case of this buffalo project, we will be looking at everything from genetic and in vitro phenotypic traits in viruses to try and help us understand what happens within buffalo that are infected with different strains of viruses, and from there up to transmission in buffalo herds and across the landscape at Kruger National Park in South Africa. And then, of course, foot-and-mouth disease virus does spill over into cattle populations from there occasionally too, and we are interested in that as well. So really, the idea here is to link process models across biological scales, and for this highly contagious pathogen, or group of pathogens rather, to ask, well, can we predict from viral genetic or phenotypic data which viral variants will spread in host populations and persist at the landscape scale? And can we identify the variants most likely to spill over into vulnerable host populations? So yeah, that really would be the Holy Grail, right? This is Monty Python's version here. And the attraction, of course, of the Holy Grail is that it's so alluring because it just always stays slightly out of reach, but we'll try. And so what we will be attempting to do here is to start at the very tiniest scale, cellular and sort of viral scale here, and use experiments in vitro to characterize a bunch of different viral strains in terms of their behavior at infecting host cells. You can then model that with a model structured like this, where you have susceptible cells here, T, then cells that have been exposed but are not yet producing virus, then infected cells down on the right here, which are producing virus actively, and of course virus in the plasma or in the medium. And so you can measure key rates in experiments, so such as infection rates, viral production rate, rate of viral decay, and then use a mathematical model such as this one to try and make sense of the things that we measure in terms of the dynamics of what's happening in terms of viral population growth and decay and cell population growth and or decay. Next scale there is the within host scale. This would, of course, look at infection experiments in buffalo with different viral strains again, where you might come up with a model like this. You could partition your model into three essentially compartments here: pathogen P, and then innate and adaptive immune responses, and look at the kinetics of viral population growth within the host. Rate, and then innate and adaptive immune activation rates. From there, then one could go up to transmission dynamics among hosts in buffalo herds, and that's probably a more familiar type of model here, where you would use, in this case, observational studies to observe transmission among animals, either wild animals or animals in an enclosure. And then parameterize from there a sort of SIR or SEIR model here, where you partition the host population, in this case, into susceptible, exposed, infected, and recovered hosts, and then maybe with a carrier or maybe not, and maybe maternal immunity and seasonal reproduction are important or not. Models are flexible, and you can try out what seems to matter most. Here's some of the key parameters that you'd need for those models, estimated from data. Well, and then ultimately you can take that up to the landscape scale, both in terms of movement of viral strains within the host population that is buffalo, or also movement across boundaries of parks and into agricultural areas. And so those are a little bit harder to draw in terms of model structure, but they're essentially landscape models that are based on herd contact patterns and maps of viral strain cross-immunity that sort of define where a given virus can and cannot invade. And so key parameters there are persistence time of a strain in a herd, its dispersal rate across herds, the breadth of cross-immunity that it provokes, and then competitive ability in carriers. So does it tend to kick other viral strains out from the lymph nodes, essentially, or does it do well at persisting even in the face of competition for that spot with other viral strains? So I'll not be talking about all of that today. I will be focusing first on familiar territory here, transmission dynamics of foot and mouth disease viruses in African buffalo populations, because that is where we started out with this project too. Then I'll move down a scale to viral and immune kinetics in individual buffalo. And finally, I'll see whether we can link those two. So can the lower organizational scale, the viral and immune kinetics in individuals, inform or allow us perhaps to predict how a viral strain might do in terms of transmitting among hosts and how it will do in buffalo herds. Right, so specifically there, I'll be looking at first FMD transmission dynamics, so how do FMDVs persist in buffalo herds? Then second, within host viral and immune kinetics, what determines viral fitness within hosts? And then third, how do within host dynamics relate to virus transmission among hosts? So you're all familiar with foot and mouth disease viruses, maybe some of you less so with the wildlife reservoir for the viruses in sub-Saharan Africa, the African buffalo. So just a little bit of background here. Buffalo are gregarious bulk grazers in savannas. They occur in herds of 30 to 1,000 animals. So they really are quite sociable, which makes them pretty great hosts for all kinds of infectious diseases, as there's usually a lot of scope for passing infectious organisms around between animals. FMD causes mild lesions in some buffalo, typically actually none. Buffalo do mount fevers and a typical acute phase response when infected with foot and mouth disease viruses, but it's pretty much inevident from the outside. Transmission of foot and mouth disease viruses is generally really efficient in free-ranging buffalo herds. So if you go ahead and test a bunch of free-ranging wild buffalo for the 3 serotypes that circulate, certainly in South Africa, so that's South African territories, serotype 1, 2, and 3, then by the time those buffalo are 1 or 2 years of age, they will have all been exposed to all of those strains. So some transmit a little faster than others. Specifically, SAT1 tends to get around faster than SAT3, and SAT2 is somewhere in between. But generally, that transmission is really, really efficient. And in adults, you'll get close to 100% seropositivity rates. The virus can be carried in dendritic cells of the palatine tonsils for months. And that's been known for a while, but until we did this project, it wasn't really clear to what degree carriers play a role in the transmission dynamics of the virus. So the first part here is published, and so I'll not spend too much time on it, but I will talk about transmission dynamics during acute infection and the role of carriers as a way to set the stage for the newer work, which is about unstable antibody titers, which may facilitate viral persistence. So where we started out was with a sort of null hypothesis, looking at foot and mouth disease transmission dynamics during acute infection, where the hypothesis is that FMDV might function as a childhood infection in African buffalo populations. So perhaps FMDVs transmit among susceptible calves, with the latest born calves of the year transmitting to the earliest born of the following year. I should mention there that buffalo do reproduce seasonally, and there's a sort of about a half-year time span when calves are born, and then a half year or maybe a little less during the dry season when there tend to be no or very few births happening. And so that trough in susceptible supply is basically what FMDVs need to get over and surmount to be able to persist in these herds. So we built a model to represent this scenario of acute transmission among calves. It was an individual-based stochastic simulation model, which we parameterized from experimental and cohort studies that I'll show you in a moment. Importantly, we did set the starting conditions to the endemic case, where most of the older animals, the adults, or really starting at 2 years of age, will have been exposed and are assumed to be immune to the infection, which is really then dependent on the new susceptible calf cohorts that come in each year. And we did allow births to be seasonal, and the model was also structured by age, or rather stage, so calf to juvenile to senescent animal. So for data to parameterize this model, we used a cohort study where we studied a herd of 60 to 70 buffalo, depending on births and deaths, which were enclosed in a 900-hectare enclosure near the Satara camp in Kruger National Park. We captured these every 2 to 3 months for 3 years to test them for foot and mouth disease virus transmission. And we also used an experimental study for the parameters in green there, where we infected buffalo with FMDVs and allowed naive animals to contact our needle-infected animals and monitored FMDV transmission there in the boma, and separately for 1 strain each of the SAT serotypes. So what did we find? Well, it actually turned out quite interesting. If you consider that foot and mouth disease viruses sort of cause similar symptoms in cattle and are considered one disease, really, But when you look in detail, they have very contrasting epidemiological parameters between the SAT strains. And so what I'm showing you here in these graphs is each time for SAT1, SAT2, and SAT3 in blue, red, and purple, respectively, the latent period here, the mean infectious period, and the acute transmission rate from our experimental data. And so what you see is that SAT1 is really a very speedy, fast transmitting virus here. It's got a short latent period. It's got the longest infectious period and the fastest transmission rate. SAT2 is somewhere in between, and SAT3 is rather sluggish. It hangs around for a while, latent between exposure and becoming infectious. It doesn't have a particularly long infectious period, and it has the slowest, it's a little hard to see in these violin plots, but it does have the slowest transmission rate as well. And so together, those parameters result in quite different values of R naught, the basic reproductive number. So R naught is the number of secondary cases that a primary infected case would generate if it was released into a wholly susceptible population. So R naught of SAT1 is quite sky high, and this is just from acute transmission at 16, SAT2, 8, SAT3, 5, still very high, but very different from one another. How does that look once we put these parameters in our model and look at persistence of these viruses? Well, basically what you see here on the left for SAT1, middle SAT2, right for SAT3 is 2 rows of graphs. On the top row shows the number infected over time, and the bottom row shows a summary of extinction time over many model simulation runs. And so what you see here is that all of these viral strains with the parameters that we got for acute transmission of infection will go extinct before a year is out. So they're not going to persist in buffalo populations based on just calf-to-calf transmission. SAT1 will go extinct the fastest because it is so fast at transmitting, so it quickly zips through the population of newly susceptible calves and then runs out of fodder, basically. The other two take a little bit longer to do so based on their slower transmission rate, but they still burn out before a year is out, or before even half a year is out, really, where the next calf cohort might come along. So none of these strains can persist in buffalo herds based on acute transmission among calves alone. Well, so next we looked at the role of carrier hosts. Since we did know that buffalo make carriers, we wanted to investigate what the implications of that are for transmission dynamics and whether that can rescue the virus from fade out in that trough between calf cohorts. So the hypothesis there is that transmission from carrier buffalo would re-spark annual epidemics in the calves. The virus would hang out in the juveniles and bridge that half a year where there's very little material for it to infect, and then re-spark the epidemic in the next cohort. So to assess that hypothesis, we needed more parameters from our experiments and from our study herd. Specifically, we needed to know what fraction of buffalo become carriers following primary infection, how long that carrier status lasts in each of our viral strains, and what the transmission rate from carriers is to naive buffalo. And that last question in particular was exciting because there had been a lot of experiments previously attempting to get transmission from buffalo, usually to susceptible cattle, and most of those experiments had shown very little transmission, either none or took a really long time. So we expected also to have to run our transmission experiment with carriers for a very long time. In our model, we extended the model by adding this carrier class here, where infected animals with primary infection, so the infectious group here, can either recover and be immune, or they can enter the carrier class for some time. Eventually they do recover, and the carrier class can potentially contribute to transmission. So we ran a second set of experiments here, using some of our animals from the acute experiment as SAT1, SAT2, and SAT3 carriers. And I apologize that the colors are different here. But basically, we did 2 groups. So we had 2 replicates there with 6 carrier animals, 2 each per SAT strain, which we allowed access to 6 naive animals. And so animals that had the opportunity to acquire each SAT strain then were 10 animals, the 6 naives, and because there's not much cross-immunity between the serotypes, our SAT1 carriers also potentially could have got infected with SAT2 or SAT3. So there are 10 animals in total for each virus to infect in each of these 2 groups. And we monitored FMD status every 2 to 4 weeks for 4 months. So surprisingly, we got transmission really quickly in SAT1 and SAT3, but none in SAT2. And so again, here we get really contrasting dynamic parameters among the SAT strains. So what I'm showing you here is on the left, the probability of becoming a carrier for the 3 strains, back to the normal colors, blue for SAT1, red for SAT2, purple for SAT3. In the middle, it's the duration of the carrier period. And on the right, we've got the carrier transmission rate. And so what you see is that SAT1 really does terrifically well in terms of both carrier formation. It has the greatest proportion of animals that become carriers after primary infection. It is actually close to 100%. It also has the longest carrier period. So that's like 250 days or so, which is definitely long enough to bridge the gap between birth cohorts. And then SAT1 and also SAT3 did show some carrier transmission faster than we expected, significant carrier transmission, but it is about 100 times slower than from an acutely infected case. So SAT2 did really poorly in terms of the role of carriers in that it was sluggish at forming carriers. It didn't have very long-lasting carriers, and most importantly, we did not observe any transmission events in our SAT2 carriers, from the SAT2 carriers to the naive or SAT1, SAT3 infected animals that we allowed them to mingle with. SAT3 had a pretty high probability of becoming a carrier, and it did transmit. So again, the take home here is that these strains really behave very differently in terms of their disease dynamics. So when we plug these parameters along with our acute parameters from before into our transmission models, what happens is that SAT1 persists robustly, given that it is allowed to have carriers. It has nice predictable dynamics, and its 10-year persistence was 100% in our simulations. SAT3 also persists reasonably well, but it's not totally reliable. So we had 10-year persistence in 60% of our model runs. SAT2 doesn't persist, which makes sense. It didn't persist previously, and since it didn't show any carrier transmission, it still doesn't persist. So carrier transmission rescues SAT1 from fade out, but SAT2 and 3 still don't persist, or at least not reliably. So what else could be going on here? And this is where the new work starts. So one observation that was really puzzling to us, and to some degree still is, was that we saw some really unstable antibody titers in our study herd. That's the cohort herd, not experimental animals. It's the herd that was confined in that 900 hectares enclosure in Kruger Park. And so the work I'm about to present on this topic is work with a graduate student, Ricardo Reyes Grimaldo, who is doing this for his PhD thesis. So what we observed was in our wild herd, when we just looked at sort of a snapshot of data initially, just sort of toying with it before we started any serious analyses with it, is that we saw way more susceptible animals across all age classes than we had expected. So what you'd expect with foot-and-mouth disease in buffalo is that you would see some susceptibles in the calves, in the 1-year-olds, maybe in the 2-year-olds still for SAT3. But after that, not really. They should all be seropositive for everything. Maybe sometimes there's a lapse, and you'd get a low rate of susceptibility in older age classes, but not a ton. And so that is more or less what we did see in SAT1 here. Sorry for the green color, but that is SAT1. And on the y-axis is susceptibility, which is just percent of animals that were not antibody protected, assuming that the level of protective titer is the same as in cattle approximately. And then there you do see that the calves and the 1-year-olds are, highly susceptible. The seroprevalence is low, and then seroprevalence is higher in the older age groups, as you would expect. SAT2 looks relatively well behaved still, although the age where you see a lot of susceptible animals goes up quite a bit higher and higher than we would have thought. And then SAT3 is really shockingly all over the place. So there are susceptible animals here quite commonly, even in adult age groups. So we decided to investigate that further and look at patterns of antibody titers in individual buffalo. And for context here, our study herd over its history would have been 40 to 70 animals, depending on births and deaths, and it had been in that enclosure since 1998. Those were buffalo caught from Northern Kruger Park. They would have come in with all strains of foot and mouth disease virus. We measured those over a 3-year period every 2 to 3 months, and actually we detected no current transmission anymore of SAT2 and SAT3 in our study period between 2014 and 17, whereas SAT2 was still circulating. So with this population size confined for 20 years, it looked that we had lost SAT2 and 3 from the herd, but recently, because we still had a lot of seropositivity, but not in the young baby calves. So those had recently been lost. And I think this is an important piece of information here because those animals were not constantly being re-exposed to the viruses, so the antibody dynamics were visible to us, where otherwise they would have been invisible because of the constant challenge with virus that would essentially boost them and make them remain positive almost all the time. Anyway, so our total sample size of animals was 144. That's higher than 70 because you have some dying, some being born, and so it all adds up to a lot more individuals. Per animal, we had on average 9 captures in those 3 years, but a maximum of 15. And again, we assumed the threshold for immune protection to be the same as in cattle. And so here's how that looks for our 3 SAT strain, well, not the experimental strains, for the 3 serotypes in our study herd. So SAT1 up here is slightly well behaved, at least towards the end of the study. So there are a lot of animals that stay above the susceptibility threshold here. That's the dotted line. But even in SAT1, there's sort of a lot of movement there between protection and no protection. SAT2 more so, and SAT3 is just an absolute spaghetti mess of animals bouncing around between high and low antibody titers. And again, these will have been animals that were exposed in the past. The bounces back up in SAT2 and SAT3 are unlikely to be from re-exposure, retransmission. They're likely to be endogenous to the animal. So for whatever reason, these animals are sometimes losing their protection and then it pops back up. In SAT1, when antibody titer is gained, it's unclear whether it's a transmission or a re-exposure event or some endogenous process to the animals. So the SAT1 data in terms of antibody gain are harder to interpret. So making sense of this with some stats, we, and by we I mean Ricardo, built a statistical model to estimate the hazard rates of antibody loss and gain for the 3 serotypes. And that's charted here. So this is the mean wait time until an event happens. And in the case above here, it's antibody loss, below it's antibody gain. And so antibody loss happens really quickly in SAT3 and less quickly in SAT2, and antibodies are actually relatively stable in SAT1. So you have to wait a long time before you are likely to get a blip down. The opposite for antibody gain. So antibody gain is quick in SAT1, maybe endogenous, maybe transmission, and it's fairly similar, but a little bit slower in SATs2 and 3, and those will have been all or mostly endogenous. So what happens when we put this additional information about immune protection being more dynamic than we would have thought into our model? So we extended the model again, and what we did is we built in another class here for lost immunity, which takes account of antibody loss. That's how you gain the lost immunity class, and then antibody gain brings animals back into the recovered class. And so animals in that antibody lost immunity class, are assumed to be susceptible while they're in there and can become infected during that temporary period of susceptibility. In terms of dynamics and persistence of viruses, then, here's what you get with acute, carrier, and unstable antibody dynamics in the model. So we, of course, still get stable persistence for SAT1. We get something very close to stable persistence for SAT2, despite its terrible performance as a carrier. And we get stable persistence and actually pretty high infection rates overall for SAT3. So SAT3 does really well with this mechanism, which makes sense given that it has the wobbliest antibodies of them all. However, it's of course questionable, given that we assumed the sort of cattle threshold for susceptibility to infection, whether the dynamics look exactly like this. And so we relaxed that assumption and allowed the susceptibility of the lost immune state to vary. So they could be 100% susceptible, just as susceptible as an animal that's never seen infection, or anything down to not susceptible at all. And that's what's on the y-axis of these three graphs here of this sensitivity analysis. So at the bottom of the y-axis, they would be not at all susceptible. At the top, at 100% susceptible, so the same as an animal that is naive to infection. And then on the x-axis of each of these, you've got population size, and we're looking at percent of the population persisting to 10 years. So SAT1, of course, has stable persistence anyway. It doesn't need wobbly antibodies. SAT3 here actually has really robust persistence even when you allow that susceptibility of the lost immune state to vary, right? So it can be down to, 40%, 30% right here on the y-axis. So those animals are not nearly as likely to become infected when exposed to virus as a naive animal, and you still get really persistent infection in a buffalo herd with this mechanism for SAT3. SAT2 is a little bit more iffy there. You need pretty decent susceptibility, more comparable to the naive animals in smaller population sizes. Once you get to really big populations, that can be relaxed as well. So the upshot there is that loss of antibodies explains persistence of SAT2 and SAT3. Definitely for SAT3, SAT2 less so, but it certainly helps. So in summary then, for the FMD transmission dynamics, how do FMDVs persist in buffalo herds? We looked at a bunch of different mechanisms here: acute transmission, carrier transmission, and antibody stability as mechanisms that might allow persistence. And so we found dramatically different dynamics among the three serotypes. Where acute transmission really doesn't lead to persistence for any of the serotypes, but the R0 of the acute transmission is really different among them. Carrier transmission rescues SAT1, antibody stability rescues SAT3, and SAT2 is still a little bit enigmatic. And interestingly, once you put all of those mechanisms into the model, the serotype that actually has the highest abundance over time is SAT3. So, so much to the FMD transmission dynamics. I will switch gears now and talk a little bit about within host viral and immune kinetics, and then try and put that together and see how one predicts the other. So to look at within host viral and immune kinetics, I'll go back to our viral challenge experiment. It's the same experiment that I mentioned before, where we needle infected some buffalo in 3 groups with a SAT1, a SAT2, or a SAT3 strain, and then watched transmission occur to naive buffalo that we allowed to mingle with them. So we measured virus and host immune responses in all of the groups on days 2, 4, 6, 8, 11, 14, and 30, so that we could get a pretty dense time series of viral population size and of host immune responses. So here's what we measured: pathogen titer in the blood. The ones you want to look at really are the pink. Those are the in-contact buffalo, the naive buffalo that were infected by the natural route by contacting our needle-infected animals, which are shown in blue. Then innate immune responses, we measured a bunch. I'm showing haptoglobin here because it performed best in terms of correlating very well with infection and having relatively predictable dynamics in each individual animal. And then for adaptive immune response, we measured neutralizing antibody titers for each of the serotypes. And so, you know, you can see some differences here among the serotypes, but really before you plug them into a model, it's quite hard to make sense of them because these are all connected, of course, right? The pathogen titer is going to provoke an immune response. And so, you know, is the titer high because the immune response is slow, or is the immune response slow because the virus was slow to get going? It's very difficult to interpret until you model the process, which is what we did. So here's our model. We modeled pathogen titer P, which grows by viral replication and is cleared through innate and adaptive effectors that the host produces. The pathogen also activates the innate immune response, I down here, and there's always a maintenance rate of innate immune effectors around. So they have their own internal dynamics, but they get bumped up when they come in touch with pathogen. And then the adaptive response here gets activated both by presence of the pathogen and by the innate immune response, sort of realistically. And it then becomes the vehicle of clearing the virus. And that work, I should mention, was led by Caleb McDonald, who was at the time a doctoral student with my collaborator, Jaire Gulbadak, at the University of Louisiana. He's now a postdoc. And so first of all, our model captures viral immune kinetics in the host quite well, and that's what I'm showing here in these graphs. So these are immune and viral kinetics in each host. On the left here for SAT1 infected hosts, middle SAT2, right SAT3, and what you see in the color is the viral population dynamics. In the dark gray is neutralizing antibodies, and in the light gray here is haptoglobin or innate immunity. And the yellow bars are when the host mounted a fever, which is quite a nice sort of reality check of what's happening there. And so the models do manage to reproduce the dynamics that we measured here, the data quite well. And you can then, from those model results, derive interpretable dynamic parameters for what's happening in the host. So first, looking at viral dynamics here, we can now compare across the three serotypes and see that the growth rate is highest in SAT1 and lowest in SAT3, SAT2 somewhere in between. And associated with that, the time to maximum viral titer is shortest in SAT1 and takes way longer in SAT3. We can look at immune responses and make sense of those. So we can look at activation rates of innate and adaptive immunity and see that SAT1 is actually quite immunogenic in terms of the innate response. It's really quick to hit SAT1 and SAT2 as well, whereas SAT3 really flies under the radar quite a bit more here. It's not detected as quickly in terms of activating innate immunity as we measured it. Adaptive activation rate is particularly high for SAT2, so that antibody response kicks in quite a bit faster, slower for SAT1, and SAT3 is sort of in between, not statistically distinguishable from SAT2. Viral fitness, then we looked at in two different ways: maximum viral load and cumulative viral load, so the amount of virus that each individual pumped out throughout the infection. So that's the area under the viral population curve. And the results here are really, really clear that the viral fitness was low for SAT2 compared to the other two and comparable for the other two. So less SAT2 virus was produced in each infection than the other two. So the viral kinetic pace and immunogenicity, viral fitness are really contrasting in each of these strains that we looked at in our experiments, where we basically have the super fast SAT1 that zips through its host, and we have a sluggish tortoise-like SAT3, but both of those contrasting strategies lead to high viral fitness in the individual infection, so high amount of virus produced per host. Whereas SAT2 plays a sort of intermediate strategy, but it doesn't work very well. So it's a playing a pug strategy. It's not a fast animal. It's also not a tortoise, but it's not very fit. So not much virus by comparison is produced in SAT2 infections. So then trying to connect the within host kinetics to population dynamics of the virus is where this gets really exciting. Ooh, the arrow. Okay. So just to remind you, we previously measured epidemic parameters for acute infection of the three strains in our experiments and models that I presented. And this is about acute infection only, this connection for now. We are still working on the carrier models for it. So what you can do, and what's really exciting, is that you can line up here parameters that we measured within the host, so viral replication rate, and then adaptive activation rate, and derived from the viral population growth and the immune responses in the host, sort of interpreted through our process model, and line those up with our epidemic parameters that we measured by watching transmission among the hosts. So here, latent period on the left, and then transmission rate in the middle, and infectious period on the right. And we can see that those are actually fairly well correlated. The latent period correlates really well to the viral replication rate. The transmission rate among animals correlates quite well to the viral replication rate within hosts. And then the adaptive activation rate within hosts predicts fairly well the infectious period of that host, which makes sense because the infectious period ends when the antibody response shuts it down. So these things make sense. But what's so exciting then is that we can look at R naught, the basic reproductive number. We can calculate that as infectious period times transmission rate for the epidemic parameters. And then infectious period and transmission rates I just showed you can be expressed as functions of the viral growth rate and adaptive activation rate that we measured within the host. And so we can ask, well, does the R naught that we estimate from these within host parameters agree with the R naught that we estimated from watching transmission among hosts in our experiments? And they do correlate. So there's a qualitative match there between the two organizational scales, which is exciting. So R naught from our within host kinetic parameters aligns qualitatively with R naught estimated from observed transmission events. So can viral immune kinetics within individual hosts predict FMDV transmission among African buffalo? Maybe. The differences in viral kinetics within hosts explain contrasting disease dynamics, but we'll need more than 3 viral strains to test this. And actually, this was supposed to be my summary slide here, which has all of the pieces. So FMD transmission dynamics, carrier transmission, and unstable immune protection play a role. And then for the immune and viral kinetics, it is a combination of viral growth rate and immunogenicity that determines viral fitness. And different combinations can work. A slow growth rate with low immunogenicity works for SAT3, and a fast growth rate and high immunogenicity still works really well for SAT1, but the in-between strategy of SAT2 doesn't work so well, and it is a less fit virus. And then how do within-host dynamics relate to virus transmission? Well, that is what I was saying in the summary slide that I should have deleted. It is that we can correlate those, and they match quite well, and that's exciting because we might be able to predict which strains transmit well at the population scale from what we see happening within individual hosts. So we'll need to investigate more strains, and that is just what we're doing with a new grant that got funded last year. And we are looking for PhD students and postdocs, so here's a shameless plug. There's my contact email to get in touch. And with that, thank you to the organizations that are helping us and funding us, and I will take questions.
Thank you very much, Anna, for this fantastic presentation. We do have a few questions on the chat. The first question is from Aldo Decker. He is asking, how does your model depend on your prior assumptions? So in his group, they've developed several models for quantifying transmission. One that uses a low model using the final size, and the other one using a GLM strategy using the time to infection and estimating the transmission rate and infectious period. In both models, he has not seen any differences between the transmission rates across the three viral strains. And now I don't see the rest of the question. So he's basically asking, do you feel that this is a model valid based on the prior assumptions?
So I think the difference there of these models compared to what's been previously done is that there are actually really few prior assumptions there. We estimate all of the parameters from data, and the big advance was being able to do these series of infection experiments, which allowed us to do that rather than estimating post hoc from infection patterns or cross-sectional patterns of seropositivity, or having to extrapolate from patterns of infection downward. And so these models are really, really well supported by data. Now, limitation is always the sample size of animals that we can involve in our experiments. And then, of course, that we really did only look at one strain per serotype. And so there is undoubtedly a lot of variations within serotypes across different viral strains. I mean, they evolve fast and they're incredibly variable. And so it is as yet unclear, whether our findings here are about SAT1, SAT2, SAT3, or whether about those particular strains that we happen to use in our experiments, and we hope to find that out. But there are not strong prior assumptions. Where priors did come in is in our Bayesian analyses, where for example, for estimating carrier parameters, we did collect all of the available previous experimental evidence for how long carrier transmission should take to occur, and used that as a sort of moderately informative prior, rather than starting from scratch when we estimated our parameters using our data. And so that's where prior assumptions come in. It's in form of a formal Bayesian prior rather than assumptions of the model per se, if that makes sense.
All right, great. The next question is from Zurin Aslin. He's asking, what is the threshold for immune protection in cattle and how to measure it in a herd?
So the threshold in cattle, and I'm going to mess up on the unit of measurement, it's 1.7, but I don't recall off the top of my head what that is like per mil. And how do you measure it in a herd? So, could we measure when a buffalo is actually protected from reinfection? So I think one way usually of measuring that is in vaccine studies, where you measure antibody titers in response to a vaccination, or you could do it by through an experimental infection as well. And then you would attempt to infect animals that vary in their antibody titers, and you see whether or not you're successful with that. We have not done that type of experiment in buffalo. We're sort of pretty limited in how much we can do experimentally. So our infection experiments were sort of pushing that to the limit, and that's not something that we've been able to address experimentally, which is why we did that big sensitivity analysis. So if our animals are a little more or a little less susceptible with the antibody titers that we measured, we were interested in seeing how that would affect our results. And it does make a difference, particularly for SAT2. For SAT3 and SAT1, it wasn't that important of a parameter.
All right. The next question is from Anna Ludi. For measuring antibody responses, what assay has been used? Is it the virus neutralization test or the ELISA or something else? If the analysis used ELISAs, has the non-cross-reactivity of these ELISAs been taken into consideration?
So both. We basically unleashed all of the available diagnostic assays on these data, since the experiments are such a rare and precious thing. So yes, we did virus neutralization assays, and we also did ELISAs, and there were a couple of different versions of them too, but I would need to defer to my colleague Eva Perez, who ran a lot of the diagnostic assays, and so if that doesn't answer the question sufficiently, then Anna, please email me and I'll connect you with the right information.
Great. The next question is from Nick Knowles. Anna, you extend your conclusions to each of the 3 SAT serotypes based on limited virus strains. How do you know that different genetic strains of each of the 3 serotypes may give different results?
Yeah, that's a great question. And I would just say exactly. We did just have 1 strain per serotype, and we don't know whether that's in any way representative of that serotype. And actually, I expect that there'd be a lot of variation in dynamic behavior within serotypes. And so that's what the work that's currently just starting aims to fill in, where we will certainly in vitro look at about 30 strains per serotype, and then in vivo we have to obviously whittle that down quite a lot. And I think we're projecting for 6 strains per serotype, where we can study within host dynamics and transmission dynamics experimentally. And then in the field, we'll look at, well, how many strains are there circulating and how are they behaving? We actually don't know, at the finer scale genetic level what all is circulating in buffalo, but we did just collect samples from a lot of buffalo throughout Kruger Park to find out. So that's a great question, and I think it's super important to try and answer that, to understand what patterns to expect among serotypes more generally than with our sort of rinky-dink little 3-strain start. But you have to start somewhere.
Great. The next question that we have is from Daniel Gisaldemisi. I think this is related to the threshold of immune protection. So he's asking whether the threshold is related to movement or biological.
I'm not quite sure what the question is. So is the threshold related to biological happenings in the animal? I think what you're asking is whether the level of protection might change according to physiological or ecological circumstances of the animal. And if that's right, then that's another piece that we're investigating right now. So we have these antibody dynamics. We can measure that antibody titers vary, but why they vary is as yet not really clear at all. And one can come up with a lot of really interesting hypotheses about that. So there could be variability with the animal's nutritional status. There could be variability with the animal's hormonal status in terms of mating season and participation therein, or parturition in the females potentially. Or there could be co-infections that play a role because they're either boosting or hindering immune responses to foot and mouth disease virus. Or there's other factors that I haven't thought about yet here. So Ricardo, the student who's doing the antibody modeling, is also doing a big statistical analysis of what was happening physiologically in our animals when these antibody fluctuations were going on. And we measured quite a lot of physiological variables and quite a lot of potential co-infections. So hopefully we'll get some more clarity there. And then also in our current study that's just now starting, looking at foot and mouth disease immunity in some free-ranging animals, we'll try and extend that further and find out. I think that's a really interesting question. And in order to predict anything with, environmental change or stress on the animals, that's certainly something we would really like to know.
Great. So Nick Knowles has a follow-up comment. He says he has studied buffalo FMD viruses over a 10-year period from all over Zimbabwe and have sequence data for about 180 viruses. So maybe you two should connect and discuss some more.
Yeah, that would be great. And actually, one of the first things we're doing in this new project is building FMD phylogenies from buffalo and from cattle around buffalo herds. And so if we were able to include more strains, then that would really increase the power that we'd have to make draw conclusions about evolutionary rates that we'll be looking at among the different branches of those lineages, and movement rates as well, geographic movement rates. So there's a whole phylogenetic aspect to this project that will be based on all the available lineages from buffalo and cattle around buffalo that could be really enhanced by collaborating broadly with people who study this in different places. So that would be wonderful.
Excellent. So I'm just gonna ask a question. In your presentation, you talk about antibody stability differences depending on which viral strain the animals are infected with. So this begs the question why antibody titers might be more stable in response to some viral strains than others. Is there anything known about that? And do we know any of the factors that may influence these antibody titers to drop suddenly below protecting levels? Is there anything in the discussion about immune exhaustion or anything related to that?
Yeah, so I don't know, and I'd really like to know why some, so I can formulate some hypotheses why some animals tend to drop their antibody titers more or less, like we discussed in the one question. But why are some strains more prone to being dropped in terms of the immune response? I really don't know. I imagine that perhaps the sort of aggressiveness of the strain as a replicator in the first case may play a role in terms of the level of antibody response that's produced to it and the, titer of memory cells that are going to be set aside to remember that particular strain. But I'm not a good enough immunologist to really understand those dynamics. And so if there are people in the audience who have more experience with understanding the differences in why some pathogens and FMDs and FMDVs in particular tend to provoke a more long-lasting immune response than others, then I'd be really interested to hear about that.
All right, very cool. Anna, thank you so much. I don't see any more questions, but I just wanted to thank you again for a fantastic talk. And we do have another question. It says, is there a difference due to different subspecies of African buffalo across Africa?
That's a really interesting question. So I've not seen any work on forest buffalo, for example, and foot and mouth disease viruses. If it's out there, I'd love to hear about it. I bet there would be differences because every host is going to respond, probably quite a lot differently, actually. And then also the ecology of those animals is going to be quite different with smaller group sizes and probably smaller home ranges. And so the whole contact structure differs among places and surely among subspecies. And at the landscape scale, that would then really affect dynamics quite a lot. But the places that I know well just have the synchronous kappa. And so I don't know how to answer that question, but I would love to know more about it.
All right. We have another question. Is there any relation between non-structural and structural protein antibody response in your study?
I'm not sure that we've made the correlation. So we did measure non-structural protein antibodies. But I don't think we've compared them directly. So, they do what they're supposed to do. They go up when they're supposed to go up. But I don't think within each individual that I've looked at that correlation. Yeah, I'm not sure. I guess I would expect them to be positively correlated, as in if an animal is, particularly fast at activating its adaptive immune response, then that should include both antibody groups, but I haven't tested that.
All right. So I think with that, I'm just going to pass it to Tessa for closing the session. Thank you again, everybody, for participating, and most especially, thank you, Anna.
Yes, thank you, Giselle. Thank you, Anna. Thank you for everybody joining. We hope this was a fruitful webinar, and we look forward to seeing you in Uganda in November.
Thanks, everyone. Thanks for coming, and thanks for having me.
Thank you. Bye-bye.
Have a great rest of your day.